Good morning, everybody. I'm Thibault Coste. I'm a molecular geneticist in the neurovascular genetic department in Saint Louis Hospital in Paris. And I'm now going to talk about molecular genetics of fetal intracranial hemorrhage, and particularly with COL4A1, COL4A2 variants, which are the most common genetic cause. Just a brief introduction about fetal intracranial hemorrhage is a serious event most often discovered during the second or third trimester. The incidence is not precisely known, but it's estimated at about 1 in 10,000 of pregnancies. The ICH can be classified in 4 grades according to the extent and location of the bleeding in the brain. And basically, the grade 3 and 4 are associated with a poor prognosis and lead to a termination of pregnancy in most cases. What about the causes? We don't know exactly about the mechanisms of fetal intracranial hemorrhage. Is it a cerebral vessels fragility, a leakness? Is there a rupture of the vascular malformation, a non-normal cerebral angiogenesis? We don't know, actually. But it exists non-genetic causes that are well known, such as fetal-maternal platelet alloimmunization, which is the most common non-genetic cause, infections like cytomegalovirus, drugs, abdominal trauma, and beside it exists genetic causes that I'm going to detail in the next slides. I made a review of the literature of all the known genetic causes in fetal intracranial hemorrhage, and as you can see in this slide, there is several biological pathway, but what is very interesting is for each biological pathway incriminated as coagulation, thrombosis, extracellular matrix, and other genes. There is very few case reports. For example, for factor 10 deficiency, only 1 case report. But for the extracellular matrix, and particularly for COL4A1, COL4A2 mutations, there is more than 30 reports of fetal intracranial hemorrhage. So COL4A1, COL4A2 is a major genetic cause in fetuses. What about the history of COL4A1-A2 and fetal data? So you all know that in 2005 there is a link between COL4A1 and the human disease porencephaly. The following years we have an expanding of the disease spectrum, and it's only in 2012 that there is the first report of a fetus. It was at 21 weeks of gestation, a fetus with a bleeding in the germinal matrix, and the following years there are more and more reported cases of fetal ICH, and all of this allowed us to clarify the main brain lesions and highlight a wide spectrum in fetuses. And also at this, there was no information about neuropathological data in fetus until 2021, and after that we could have the information that ischemic and hemorrhagic lesions are present with various abundance and age. What are the MRI and neuropathological data in COL4A1 fetuses? So here is some example. In fetus, we could have multifocal ischemic and hemorrhagic lesions. Lesions are of variable ages and abundance, and could be associated or not with the porencephaly, as illustrated there is the cavity, and our schizencephaly, the communication between the ventricle and the interhemispheric space. And about the neuropathological data, we have some information. So we observed also multifocal ischemic and hemorrhagic lesions, but we can find some petechias in the brain parenchyma. There's also marker of hemorrhage, such as iron-rich macrophages in the ventricular zone, numerous hemosiderin deposits, in particular in ependymal and supraependymal regions, and reactive gliosis. All of this allowed to delineate a wide spectrum of brain abnormalities in those fetuses, with the porencephaly, which is one of the main sign described in literature, maybe because there's a bias, because initially people with porencephaly were tested for COL4A1, COL4A2. But we could have also ventriculomegaly, schizencephaly, calcification, polymicrogyria, periventricular nodular heterotopias, anomaly of the corpus callosum, of the cerebellum, and also a accumulation. Another important thing is that it exists a strong intrafamilial variability and also an highly variable age at onset, as illustrated here. It's an example of a family with a deletion in COL4A2 gene, and there is 2 fetal cases here with an intracranial hemorrhage, and they carried a mutation inherited from the father who were symptomatic with hemiplegia, porencephaly. The mutation were inherited from a grandfather asymptomatic, but with brain lesion on his MRI. The aunt is asymptomatic. So there is a very strong intrafamilial variability, and so this is very complicated for the genetic counseling. After this, we raised several questions. So first question was, what is the prevalence of COL4A1-A2 variants in a large series of fetuses? The second question is, does it exist differences between variants in fetuses and patients with an adult onset? Because in the adult, the onset is about 40, 50 years old with leukoencephalopathy, aneurysm, hemorrhagic and ischemic stroke. But we don't know why it's more severe, more earlier in fetuses than in adults. And the third question, the major question, does it exist novel genes involved in COL4A1-A2 non-mutated fetuses? This is very important to allow the genetic counseling, to define the genetic architecture of ICH, and to identify potential therapeutic targets. So in France, we have a big cohort of fetuses because we are a national center. We received 216 fetuses from 2010 and 2021. They were referred for targeted gene screening of COL4A1, COL4A2 after exclusion of other non-genetic causes such as fetal-maternal infections, etc. And we could identify mutations in 39 fetuses. It's about 20% of fetuses had a mutation in those genes. I put in this slide the location of those mutations in those fetuses. As you can see, it's mainly missense mutations. In blue are missense mutation, and in red are disrupting mutations. So it's mainly missense mutation and glycine in the triple helix. But for COL4A1, we could also find some mutation in the NC1 domain. And what is also very important is that there is more mutation in COL4A1 than in COL4A2 in fetuses. For us, it was blindingly obvious that the location of mutations in COL4A1 were more in the C-terminal part of COL4A1. So I'm going to show you the next slide on that. Another important finding is that 70% of fetuses had a de novo variance. So this is a very important fact for the genetic counseling. And this ratio is more important than in adults. It also implies that 30% of variants are inherited from an asymptomatic parent, and so it illustrates the incomplete penetrance and suggests the existence of modifying factors that we still don't know. The main features of our COL4A1-A2 mutated fetuses cohort, the time at discovery was 27 weeks of gestation. It's about the end of the second trimester and beginning of the third trimester. The time at termination was 30 weeks of gestation. And about the cerebral lesions, it was essentially multifocal ischemic hemorrhagic lesions, but we could also have a ventriculomegaly in about 60% of fetuses, a porencephaly in 42%, schizencephaly, polymicrogyria, cerebellum anomaly, and corpus callosum anomaly. What are the implications of the identification of the pathogenic COL4A1-A2 variant in a fetus? There is an importance of a non-invasive worker for carrier parents with the existence of preventive measures and genetic counseling. It's also very important to communicate the information in the relatives because they could benefit of the preventive measures and the genetic counseling. And a main question in fetus is how to manage future pregnancies. The question of the pre-implantation, prenatal diagnosis, the question of the type of delivery for a carrier fetus, for a carrier mother, whatever the fetus status. There is no recommendation about the management of the future pregnancy in those cases. It's just always a case-by-case discussion, I have the impression. I want to come back about the nature and location of pathogenic COL4A1-A2 variants in fetuses compared to those observed in adults. And I made a figure where I put the variants observed in adults, those are in blue, and in red, those observed in fetus. And as you can see, in adult variants are located all over the gene, but in fetus there are more clustered at the end. And so it's a difference significantly proved. And it's, we have, with this data, proven the information was already present in literature that was observed in mice by the team of Gould, where they can demonstrate that in the C-terminal part in mouse model there is more intracellular accumulation, more cell cytotoxicity, and more hemorrhagic lesions. So we have confirmed that in fetus those severe phenotype are due to the location of variants or mutations. The next part of my talk will be about the negative fetuses, because 20% of fetuses are mutated, but there is also fetuses with intracranial hemorrhage, and we don't know why they have this hemorrhage. So we set up a project to identify novel genes in those negative fetuses. We could include and perform the whole exome sequencing in 113 index fetuses, including 35 trials, meaning index and parents sequenced, 3 families with 2 affected fetuses, and 75 probands. I'm not going to detail all the data on this course because we identified several genes, but I have picked up some kind of genes that are very important and very interesting for me. We identify mutations in genes involved in hemostasis and in inherited platelet disorders. We found in one fetus biallelic pathogenic variants in the protein C gene. This fetus, as you can see in this figure of this brain fetal MRI, a massive intracranial hemorrhage, and on the neuropathological examination, we could observe numerous organized thrombosis of vessels in the white matter and the presence of hemorrhages at the surface of in the ventricles. The protein C gene is associated with 2 conditions in human. One is it's a dominant condition with heterozygous mutation leading to hereditary thrombophilia with patients with a susceptibility to thrombophilia. And another one is a recessive disease with a severe protein C deficiency. In that condition, there is neonatal cases with a severe deficit. And we could conclude that in neonatal and in fetus, ICH is due to vascular weakening following the formation of microthrombi in capillary vessels. Next genes we have identified as cause of fetal intracranial hemorrhage is MPL and MECOM. There are 2 genes involved in early megakaryopoiesis. It's 2 familial forms. The first family is biallelic mutations in MPL, and MPL is encodes the receptor for the thrombopoietin, and so patients with mutation in those genes have a severe thrombocytopenia. MECOM is an oncoprotein, but it's a dominant condition, and patients with heterozygous mutation have also a severe thrombocytopenia. And so for those fetuses, there were severe thrombocytopenia, which have not been seen at the beginning, but with whole exome sequencing, we could identify the causes. And so we conclude that in fetuses, we have to screen those genes because we could miss this kind of conditions. And the next part of genes that we have identified is mutations in genes encoding tight junction proteins. We identify a new causal gene, which is ISAM. ISAM encodes an endothelial cell adhesion molecule. It was a collaborative work. We could gather 8 families thanks to Gene Matcher. It included 13 individuals and 4 fetuses. All patients had homozygous loss of function variants, and all patients were consanguineous. I encircled our family in our cohort. There was, in fact, one fetus, and the relative was a young child. These are the brain MRI in all these patients, and all these patients have antenatal ICH. And some patients could be born, and so they develop neurodevelopmental disorders. And our patients were patient 6 and patient 11, and as you can see in the brain MRI, there is a massive destruction of the parenchyma. The ISAM is a gene involved in the cohesion of vascular endothelial cells in the blood-brain barrier, and so a biallelic variation of ISAM could impair the tight junctions of the capillary endothelial cells, leading to intracranial hemorrhage, and the intracranial hemorrhage and the impairment of tight junctions proteins lead to severe neurodevelopmental disorders in surviving children. What is very interesting is that we compared this new phenotype with the phenotypes of all the other one described for all the tight junction genes, and only for GEM3 and ISAM there is intracranial hemorrhage. And we don't know why with GEM3 and ISAM there is intracranial hemorrhage, and with the other tight junction molecule we don't have intracranial hemorrhage. So there is still work to do. So in conclusion, here are the current genetic architecture of fetal ICH. There is several pathways involved. They involve vascular stability, vascular homeostasis, like angiogenesis, basement membrane, tight junction, platelet homeostasis, coagulation, ubiquitin proteasome system, immune response. In this figure, I made genes reported in literature that are in black, genes identified in our fetal cohort, genes that are causal in blue, and in green, those that are candidates for us, but we are still collecting some information to very incriminated there. And the second conclusion is that it exists a genetic heterogeneity with multiple causal and candidate genes. There is low, even no recurrence for some gene. And it also exists some similarities with mutations of COL4A1, COL4A2, and some mitochondrial disease that I have not shown you there, but there is some fetuses who have mutation in mitochondrial genes, and they had some similar signs as observed in COL4A1, COL4A2 patients. What is very also important is that in 75% of fetal cases, we could not identify a causal genes, but there is also a large number of possible candidate variants, so there is a lot of work to do. And what are the other genes responsible of a fetal intracranial hemorrhage? What are the strategies to go further? We are still working on that. And one strategy we are doing right now is a matching strategy. We match the possible candidate variants in our fetus with genes that are associated with ICH, with vascular malformation or angiogenesis defect in mouse on zebrafish, just to see if there is some correlation. And I want to thank the institutions, finding all participants and collaborators who supported this work. They are listed there, and I want to thank you for your attention.